Introduction. The Scanning Electron Microscope (SEM) is a high-power image to the user a focus beam of electron to project detailed image of sample surface and microstructure. It detects signal like secondary and backscatter electron to create high-resolution image. SEM is widely used in material science, nanotechnology, biology, and forensic science to study surface feature and nanostructure. Researchly is used as aspect to environmental research and nanomaterial analysis, such as examining packaging wires and biological interaction. Accurate SEM image depend on proper sample preparation, include fixation and dehydration to preserve surface and core structure.
Sampling process. First, electron beam generation. A high-energy electron beam, 100 until 30,000 electron volts, is created usually using a thermal source. Second, beam focusing. Special lenses shrink the large electron beam to a tiny size less than 10 nm. This small beam interacts with the sample, reaching up to 1 UM deep to produce signal. Third, scanning machine. Scan coil move the electron beam across the sample surface in a raster zigzag pattern. Higher magnification means scanning a smaller area. The working distance from lens to sample affects magnification and is adjusted automatically in modern SEM. For signal detection, signals such as secondary electrons and backscattered electrons are detected. These signals depend on the voltage applied to the detector and help form the image.
Alright, continue with the SEM process. Let me explain what happens after the signals are detected. First is image formation. The signals that we captured earlier are now displayed as an image on the screen. We can adjust the brightness and contrast to make the image clear. And if we want to see really tiny details, we use high magnification, something like 10,000 times or more. Next is the effect of electron voltage. If we use low voltage like below 5 kV, it's good for viewing surface details, but if we use higher voltage between 50 to 30 kV, the electron can go deeper and show what is inside the sample. Lastly, we have topography and contrast. This is what gives SEM image that 3D-like appearance. By tilting the sample, usually between 50 to 70 degrees, we can increase the number of signals collected, which help us see the surface features more clearly.
Here are the simple steps for preparing biological sample for SEM. First, we fix the sample using 2.5% glutaraldehyde for 2 hours. Then, we use 1% osmium tetroxide for 1 hour to protect the cell membranes. Second, dehydration. We dehydrate the sample through a series of ethanol steps, starting from 10% up to 100%. Each step takes about 5 minutes.
Following the dehydration process, the sample is air-dried for a duration of 3 to 10 hours. Thereafter, it is placed in a vacuum chamber without a lid to ensure complete removal of residual moisture and to maintain the structural integrity of the specimen. This preparation protocol effectively preserves the cellular structure, allowing for detailed and reliable observation of the interaction between nanomaterials and the cell surface under scanning electron microscopy.
Analysis technique using the scanning electron microscopy (SEM). SEM is a powerful tool used to analyze the surface morphology and microstructure of materials at a submicrometer scale. It works by producing a focused beam of electrons that travel from a point in a vacuum, which interacts with the sample surface, producing a variety of signals that form highly detailed images. SEM is a combination of an electron column and an electronic console with controls for adjusting magnification, focus, brightness, and contrast. It can view and analyze sample surface topography, morphology, and composition, and crystallography. When integrated with energy dispersive X-ray spectroscopy (EDXS), SEM enables detailed chemical composition analysis. This combined techniques was applied to study light packaging waste materials, including polypropylene and polyethylene terephthalate. A low vacuum setup was employed to decrease the effects of surface charging without the need of conductive coating and preserving the sample for further analysis. By providing critical insights into the physical and chemical characteristics of packaging waste, SEM EDSS proved invaluable for optimizing recycling processes and energy recovery strategies, aligning with circular economy goals.
Okay, the next one is result/data analysis. The first article is Biomedical SEM conducted by Minety and others, which introduced an alternative approach to traditional SEM sample preparation. The researchers employed air drying in place of the conventional critical point drying method. Their results indicate that cell morphology, specifically shape and size, remain consistent with the traditional technique. Notably, a mixture combination of iron and cobalt nanowires remain attached to the cell surface even after multiple washing steps, suggesting stable nanomaterial cell interaction. This method presents a rapid, low-cost solution for analyzing cellular response to nanomaterials with significant implication for biomedical research.
The second article shows how SEM EDXS was used to study plastic packaging waste from the UK, France, Italy, and Romania. It found big differences in elemental composition. Romanian PP had 95% carbon, while UK samples had 71.1% with extra elements like aluminium and calcium. SEM imaging showed surface changes, texture, and particle inclusions, which were analyzed using spot mode EDXS. Compared to CHNS or analysis, SEM EDXS offer better detail and accuracy. These findings highlight the need for improved recycling methods following the circular economy ideas. The third article reviews how SEM is used to study minerals, focusing on tools like SEM EDX, SEM MLA, and QEM scan. SEM produces detailed image, detect material pairs, and identifies elements using EDS. AI and machine learning are making SEM analysis faster and more accurate. The RISE method, combining SEM with Raman imaging, help identify minerals and organic material in place. Challenges include sample preparation, EDS limitation, and high cost. Future improvement may include automation and space application.
In summary, a scanning electron microscope (SEM) is an instrument that enables very high magnification viewing of the surface morphology and microstructure of a material. Using a focused electron beam, SEM generates surface images of a specimen. SEM has widespread use in biology, material science, and forensics for investigating surface details, detecting defects, and analyzing microstructure. Aside from enabling qualitative and quantitative analysis, there are some benefits of this technology, such as the digital imaging and data processing. SEM has proven to be a handy tool despite the exorbitant costs and challenging equipment procurements due to its accuracy, broad applicability, and capability to provide in-depth information regarding the research sample.